absolute pleasure today to welcome Dr. Rudy Swart. Rudy Swart is a postdoctoral research associate with Nelson Mandela University. His research on insects in the Southern Cape Forest started in 2014 in the Garden Route National Park. He obtained a PhD looking at the diversity of canopy insects in relation to tree species in our forest. And for the past 4 years, he's been actively doing research in our forests. In my mind, brilliant scientist and definitely the future of forest research conservation. Rudy, without further ado, I'm handing over to you. Thank you.
Thank you, Henry. That was a very nice introduction. So, Rudy Swart's my name. Just before I start, this research that I'm presenting today was funded by the Rufford Foundation, National Research Foundation of South Africa, and also Nelson Mandela University. And it was done in the Garden Route National Park, so South Africa National Parks, of course, also has quite a lot to do with this research. Okay, everything I wanted to say with my intro, Henry has covered, so that's great. So, in short, insect pollinators in our forests. What do we know about it? Nothing really. A quick background, these forests that we have in the Southern Cape are termed or classified as Southern Afrotemperate forests or African temperate forests. It's the largest forest in the whole of Southern Africa in that red block over there. Although these forests, you shouldn't view them in isolation. There are linkages between these forests and forests up to the equator on Mount Kenya, for example, even into Ethiopia. Small pockets in Angola contains Afromontane forests. Many of our tree species that we have here are actually tropical, Afrotropical lineages that moved down during prehistoric or during times when the climate was different. Africa shifted northwards, there was upheaval and upliftment. But what we see today is classic remnant patches of a once more widespread forest biome. Just interestingly, we also have some classical temperate or southern hemisphere lineages in our forest as well. For example, the tree species Royals or red alder, which has its closest relatives on the island of New Caledonia in the Pacific Ocean, and also Witals, which is in the same family, Canoniaceae or white alder, who has its closest relative on the island of Tasmania. So, presumably ancient lineages of tree that still survive or remain in our forests. If we look at this map, I looked at 6 Afromontane or African mountain forests from Mount Kenya all the way to where we are today. And what I noticed shortly, I noticed a lot of things, but of the 90 or so tree species and large shrub species that we find in Southern Cape forests, 27 of those species can also be found on Mount Kenya. Now, as you would know, it's not a continuous belt of forest. There's Miombo woodland, there's savanna, there's the arid Limpopo Valley. So the chances that there's current dispersal happening is slim, although it's something I'm up to debate. But there's one species that we probably won't debate much, and that's Trichucladus ellipticus. So we have two species in the genus Trichucladus, Onderbos, which is quite common in our forests. The one is Trichucladus granitus, which we find abundantly in our forests, as you would know, Sky. Trichucladus ellipticus, there's one remnant population close to Diepwalle, north of Knysna. It disperses its seeds via ballistic mechanisms, so it's not abiotically dispersed by birds or by mammals. But that species is in the Southern Cape and it's also on Mount Kenya. So how did those seeds get there? So it's hypothetically, at least, if you don't want to take my word for it, because these forests were once much more widespread. So that's just the background context to these forests. We shouldn't view them in isolation. So even though the largest forest in Southern Africa is in this area, the Southern Cape, we also find a lot of small patches like this in the Southern Cape landscape. So forests that are technically islands within a fynbos matrix, or a heathland, shrubland, fire-driven biome. So in the Southern Cape, naturally, without human intervention and agriculture and urban development, we see two biomes occurring there naturally. forest and fynbos. And many of the forests occur in these small patches. So in realizing this, I became interested in these forest patches and specifically how the trees reproduce in these patches. Are the trees, in terms of their pollination requirements, isolated in these forests, or are they not isolated in these forests? If we imagine, if we can sort of, without thinking too deeply about ecology, you can imagine that a larger forest and a well-connected forest will have more insect species and pollinator species compared to a small isolated forest in the landscape. I'm not saying that is the case, I'm taking you on a wrong path here actually. But anyway, these thoughts came to mind. How do the trees reproduce in those small isolated pockets? Also, which insect species pollinate our trees? It's never really been studied. If we look in the literature, it goes as far back as 1926, and that's basically where it stops, by a guy, John Phillips. That's almost now 100 years ago, believe it or not. So we can't speak about the 20s anymore because we are in the 20s. Regardless, in 1926, the opening remark is there's practically no information concerning the biology of flowers, fruits, and the young regeneration of the more important forest trees, he goes on to say. This book came out in 2021, so fairly recently. It's divided by biome, and if you turn to the forest biome section and the pollinator subsection, I can just quickly read for you because it's quite small. But although there have been no detailed studies of pollination in southern African forests, there is broad agreement amongst botanists and entomologists that generalist insect pollinators are probably the most important. What is generalist insects? Is everyone familiar with that term? If we say generalist insects, so think of if you have a flower with a very deep floral tube and you have a moth with a very long proboscis, it's only that moth that is able to pollinate that flower. That's a very specialist or specialized interaction. Compared to a small flower that is open, not a deep floral tube, hundreds of species might visit it and successfully pollinate it as well. That would be a generalist interaction compared to a specialist interaction. So the prediction is that generalist pollinators dominate these forests. So my broad aim when I started this research was how does anthophilous, which is basically just a term for flower-loving or insects that frequent flowers, it's a term that sums that up. How does their diversity organize itself in this naturally fragmented but also multi-layered habitat? And I had some objectives to get to this aim, and that is, does forest type, meaning these small patches that I showed you on that photo versus these large continuous forests like Diepwalle and Gona, does forest type affect anthophilous insect diversity? Does height above forest floor impact on anthophilous insect diversity? And then thirdly, does species of tree impact on pollinating species diversity? I mean, if it's generalist like they say it is, then species of tree shouldn't really have that big an effect. Then a fly can, or a bee or a wasp can just go for any odd flower. It doesn't really matter. And this is important because there's about 90 species, as I said earlier, and if we don't know what pollinates them, how will we understand in the future if we see less recruitment, for example? Is it pollination limitation or is it something else? So it will give us an important baseline to work off of. What does the flowers look like in these forests? Very simple graph, but you just have to look at the left-hand bar over there. That's the warm temperate forest or the southern Cape forests, and that is proportion of species divided into flower color. And you can quite clearly see that the majority of trees in these forests have small, well, not small necessarily, but in terms of color, white flowers. But they're also quite small as well. So white, dominant flower color, we have a little bit of pink, like Cape chestnut, red, Burchella bubalina or Alleria lucida, which are actually bird pollinated, but white is the dominant color. So when I came up with the methodology, one aspect of this research, we used these white, these white buckets in different layers within the forest. And then the white bucket is basically a substitute for flowers and insects that frequent flowers or are attracted to white within this forested habitat will then be attracted to this white, and they will get stuck in this soapy water. So you put in a bit of soap into the water just to break that surface tension so the kochakis can fall into the buckets. So these buckets were placed in continuous forests and also in these small forest patches within the fynbos landscape. They were placed over 2 seasons, spring and summer. And the reason for that is because that's when the majority of trees flower in this landscape. There's obviously exceptions like forest elder or Nuxia, that's a winter flowering species, but apart from a few exceptions, most species spring and summer. And we placed a total of 600 of these buckets, so it's quite a lot just to make it robust and solid, and that no one can tell us our results are shoddy. So, just to quickly show you a map of where we worked, where I worked. I had 10 sites, so the C1 to 10 are the continuous forest sites. So there's Naisna. For those of you that know the area quite well, this is our big Palle over here. There's Komsepak. That's Gona of the Blanchdraai at Gona. Then you have Leerliflei, and you go to Jubilee Creek, Goutveld, Millwood area. And then the P1 to 10 are these small forest pockets within the fynbos matrix. So this is more or less the study site. The patches were on average 90 hectares in size. The continuous forests were much bigger. So for that 711, I used roads as edges, but that's misleading. In actual fact, this continuous forest is about 25,000 hectares in size, apart from the odd road or forestry road or hiking trail that cuts through it. So massive difference in size between these small pockets and the continuous forest. Okay, now I want to ask the audience, what do you expect? Just if I have to ask you a quick question, where would you find the most pollinator diversity? In the patches or in the continuous forest? And anyone can venture a guess. I want to say there's no wrong answer, but there is obviously a wrong answer.
The big forest.
Okay, we've got big forest there. Purpose? Okay. Okay, that was the wrong answer.
It's a trick question.
It was a trick question because earlier on I took you on the wrong path. I actually told you I was taking you on the wrong path, but species richness top graph. So that's the average species per bucket. In the patches it was 11 to 12 species per bucket in the patches, and in the continuous forest it was about 7 to 8 on average. So more species in the patches, and for abundance also significant increase compared to the continuous forest. Now I don't know if these questions to the audience are going well, but why might this be the case?
You catch us once, you don't catch us twice.
Maybe I can ask him again. Once bitten, twice shy, I guess.
My guess would be the surrounding fynbos matrix, maybe.
Thanks. Yes. That's at least my hypothesis. The equilibrium theory of island biogeography, I don't want to bore you with this, but it's very simple. If you imagine an ocean with differently sized islands, the larger island will probably have more species than a small island that's far away. That's quite sort of logical. What these results show us, if you sort of extrapolate it into the theoretical literature, it does not necessarily apply in forests that are naturally fragmented. So remember, in the beginning of the talk, I mentioned that these forests are naturally fragmented. It's not because of human activity. So if you go into the Amazon, for example, where a lot of fragmentation studies have been done, they literally find that smaller patches, less species, larger patches, more species. But that's because of human activity. They cut these forests into smaller and smaller pockets, and the animals that live in these forests, or the insects, what we're talking about, they're adapted to large continuous forests. So now all of a sudden you have edge effects, it's unnatural, the matrix surrounding these small forests are transformed, so it makes sense. But in the Southern Cape, our naturally patchy forests, if they're not within a plantation, pine plantation setup, they're mostly nestled, or the ones I looked at least, are nestled within a native habitat matrix, the fynbos. And this is of course a mega diverse biome, and it's globally renowned, it's a biodiversity hotspot. Fynbos supports upwards of 8,500 plant species compared to forests in this region, which supports about 470 plant species. And this article, Proctors and Cowling 2006, they looked at 4 biomes, and I think it was grassland, thicket, something else and something else. They wanted to see if plant species richness tracks insect species richness, and it does for all 4 biomes. So if you have more plants, you probably have more insects. So if we can assume then that insect species richness is more in the fynbos than the forests, we could predict that there's spillover of insect species from the fynbos into these smaller forest patches that are, some of them are as big as this room that we're in, maybe a bit bigger. What would we expect to see if there is spillover from the matrix? We will see that proximity to edge might have an effect. So even in the continuous forest, as you move closer to the edge, you might see an increase in species richness. So these buckets were placed on a range of distances from the edge. Some of them were closer to the edge, and then some of them were deep into the interior. Some of them were more than 1 kilometer into the forest interior. And if you graph this out, that's not the right terminology, but regardless, so if we just focus on the top left graph, on the y-axis you have the amount of species. On the x-axis you have distance to edge. So if you are close to the edge, you have more species, and as you move towards the interior, the species gradually starts dropping out. So you get less and less species as you move deeper into the indigenous forest. Something that's very interesting on these graphs though is we accounted for height. You remember we placed these buckets at different heights. So you can see deep in the forest, over here, you have high species richness in the top layers, and then intermediate in the middle layers, low levels in the lower layers. But as you move towards the edge, you can see that these lines sort of move closer to one another. So that's for overall anthophilous insects. The one group that really stood out were the hymenopterans. This is bees and wasps. So in the interior, you have your 3 height levels. So let's say you're standing here, you're deep in the forest. Down here, you will have low levels of species richness, and it gets more the higher up you go. But as you move towards the edge, everything increases in species richness. So the spillover is probably not only into the canopy, but also into the lower layers of the forest as well. Any questions on this graph? It's not too confusing. It's okay. So, forest type does have an effect on pollinator diversity, but also height above forest floor. I don't show the height graphs, but there is a definite height increase. So what we sort of deduce from these results is that due to the edge zone influence, that clear vertical stratification that you see in the interior is disrupted nearer to the edges. And what we predict, but this is to be tested, and we have a student that's now looking at the fynbos side of things, to compare the fynbos insects with the forest insects. But we're predicting that generalist insects, so insects that go for any flower actually, are able to utilize floral resources in the fynbos and in the forest. And this drives this heightened number across the ecotone. And secondly, insect biodiversity is both vertical but also horizontal when you're in the forest. Okay, and then the third objective was species of tree. This was done, so the study was done here, the first part, but this one was done in Cape Forest, which is also a large indigenous forest close to the George campus. And in this forest, I accessed 24 individual trees of 4 species. This was Aphrodite immediata, or white pear, Philodendron or saffron, Curtisia or assegai, and then Ilex mitis or Cape Honeybee. So these 4 species, you can see that they have those small white flowers that I spoke about, like most forest trees have, but 2 millimeters, so the saffron flower is, it's about 4 millimeters more or less, compared to assegai, that's also 2 millimeter size bar, very, very small flowers, like very small. Compared to Ilex mitis, that's 3 millimeters, and Aphrodites, 4.5. So you have size differences. And also, if you look closely, you can see that it's not the same type of white, okay? This is a recent wedding dress. This is wedding dress from the 60s, okay? Different shades of white. But that's just anecdotal. It's not been tested. From an insect's vision, at least. So this is what it looks like up in the tree. There's a lot of branches. I had my net with me, and a data sheet, and something to measure the temperature and the wind speed and all of that.
How did you get up there?
Ropes.
Roads.
Yeah. And then I'll be up in the tree while the tree is in flower, obviously, and then just wait for hochakis to visit the flowers. What insects are pollinating our trees, right? And for these 4 species of tree, 105 species of insect visited the flowers. It's quite diverse, from 50 families and 7 orders. 14 of these species were Syrphidae. Now, Syrphidae is a family of flies that we call hoverflies. They hover in front of flowers, almost like hummingbirds. So 14 species is quite a rich diversity. 2 of them weren't described yet. They were described end of last year as new species. 38 Diptera species or fly species. Diptera is just the order for flies. 26 Lepidopteran species. So Lepidoptera are your moths and your butterflies. So of that 26 species, 23 of them were moths and 3 of them were butterflies. Now you're going to say, how did you see the moths? So I also went up there in the evenings. If we look at visitation rates of the different taxonomical groups, it becomes quite clear that Apis mellifera capensis, or the Cape Honeybee, is very dominant. About 57% of all flower visitations were the Cape Honeybee, which is a huge number. If you look at the second highest species, it was 5%. So it's like from 57 to 5, and then it just goes less and less and less and less. So there's one species that really dominates in this canopy layer. And this is what the interaction network looks like. You can see that here you have your 105 insect species, and at the bottom you have your 4 tree species, but the red is the Cape Honeybee. You can see how it dominates.
What are those trees, 3 and 4?
So that is Aphrodite, that's assegai, saffron or Philodendron, and Ilex mitis or Cape Honeybee. But you can see like Aphrodite and Ilex mitis, which has the more modern wedding dress white, has wide bars, so a lot of interaction compared to assegai, which has these small, almost yellowish flower, very little interactions there. Saffron also a bit less. So it does seem, just like anecdotally, objectively, that flower morphology has an effect on the interactions. However, it does seem to be quite generalist, like we expected. Like there's a lot of insects and they overlap between the trees that they visit. I'm also taking you on a wrong path here again with the generalist thing. I'm talking to you specifically.
On the track.
We also looked at pollen. So a representative number of insects were sampled with the net up there, taken to the lab, and we extracted the pollen from their bodies. And then what we did was, and this graph looks very simple, but we actually have to concentrate. We wanted to know that insect that visited, let's say, assegai, is it carrying assegai pollen on its body or not? And is it carrying pollen from other species or not? So if we look at the black bars, so here we have Aphrodite, assegai, saffron, and Cape holly. You can see that for Aphrodite, 90% of insects that visited Aphrodite also carried Aphrodite pollen. For assegai, it was just over 70%. For saffron, it was almost 100%. So he's doing quite well, it seems. But then for Ilex metis, it was less than 50%. So ignore the white and the grey for now. If you just look at these black bars, why would Ilex metis have this dip in insects that actually carry Ilex metis pollen? Okay, so these three trees are bisexual, and this tree has male and female separate trees. So I think that's the reason why. Because I climbed both male and female trees, and for these ones, obviously, I climbed the bisexual trees. So it's much easier for pollen to get back to the tree. Imagine a bee foraging in a saffron where the flower has both male and female reproductive parts. So that increases the chance that I'm going to get male pollen on that bee. Compared to Ilex metis, if I climb in a female tree, there's no pollen in that tree. It has to rely on a male some distance away. So this might just be reflective of that I also climbed female trees. Yeah, the other bars, I'm just going to leave it at that for now. But essentially, okay, no, I can't leave it there because I have to explain the white bars as well. So the white bars indicate how many insects or proportion of insects carried pollen from other trees in the landscape that weren't that focal tree that we were focusing on. And you can see that these insects, yes, they carry pollen of saffron when they visit saffron, but they also carry pollen from other trees as well in quite high numbers. So again, linking up to the previous graph of interactions, it seems to be generalist that these insects are visiting numerous trees in the landscape and not just one. So it's not that specialized. So generalist as predicted, as with in ecology, not necessarily. For Aphrodite demediata, there were 3 species of bee that preferred its flowers and didn't really go for others. If it did go for other tree species in very low numbers, there was a species of Pipinculidae, which is a big-headed fly. I'm not mocking him, that's the official term. It does have a big head. Exclusively visited the flowers of assegai. It didn't in high numbers, so it's not just like it visited one and then it's 100% because it's a single visit. It visited assegai recurrently and not the other species. So that can be seen as a specialist. There was a species of hoverfly that only visited saffron flowers, and there was some wasp species that really loved Ilex metis again. So even though it's a generalist system, as predicted by other authors, there seems to be some assembly going on. And this graph shows it quite clearly. So on this graph you have different dots or symbols, where each symbol, the closer it is to another symbol, the more similar the community of insects were that visited during that session. So each symbol here represents a session that I was up in the tree. So let's say those 2 blue ones are close to each other. This is just, I'm just talking now nonsense, but let's say there were 3 species of bee that visited the flowers 5 times each, 15 interactions for 3 species. Now the blue dot right next to it probably had the same 3 species, 5 visits, 10 visits. So now it's up to 20. So it's very similar, but it's slightly different. So the assemblage of visitations are slightly different, but the dots will be quite close to one another. If you have a dot that's far away like that, there were probably one of those bees visited it once, and then there were fly species and beetle species that didn't visit the blue dots at all. And if you plot this graph in terms of similarity of the sessions, and you color code it by species, so blue is Aphrodite, green Curtisia, and so forth, you can see that the species of tree does sort of separate on this discriminant analyses. And I don't want to bore you with stats, it bores me, but significantly so. So these differences are significant. What it tells us is each tree has a unique community of insect visitors. Yes, there's overlap, but one might go to assegai 10 times, but only goes to Aphrodite once or twice. So it likes assegai a bit more. This is exciting. We measured the petals and the pistils, so we could see from that previous photos some flowers were smaller, some were larger, but we had to sort of statistically prove it. That's assegai, small flowers, larger flowers for Aphrodites and ilex. We also had what was the thing called? A spectrometer, where you, in a dark room, you measure the reflectance of the petal, the whiteness, so you measure the whiteness of the petals, and they too separate in terms of insect vision. So some insects, or they will view assegai, assegai and saffron, they will view quite similar. It's sort of an off-white, yellowish color, whereas Aphrodites and ilex separates. It's especially for bees, it's much more visible. So generalist, specialist, well, Apis mellifera, the Cape Honeybee dominates, okay? Phillips also found that in 1926, and that's why I believe this is a natural phenomenon. It's not because of beekeeping in this area. Also, Groenkop, where I work, to the north is fynbos, to the east is more forest. To the south, there's a farmer there, but he doesn't have bee nests on his property, and to the west is campus, which I also haven't seen bee nests on our campus. I also found a bee nest natural within a yellowwood in the study area. So I think this is a natural phenomenon. How successful these Cape honeybees are in terms of pollination is a different study altogether. They visit them in high numbers, but whether or not they're successful pollinators, we don't know. Despite both trees and insects being generalist, as we predicted, some insect species show strong preference for certain species of tree. So it's generalist, but it's not random. It's not necessarily opportunistic. It seems to there's a deterministic aspect to it. And this community disparity that we see between the trees might be explained through aspects like flower size, flower color, but perhaps even scent or nectar rewards, stuff that we didn't look at. I wonder if I'm done now. I'm close to being done. This is now interesting. So how do I link the tree climbing with the landscape stuff that I showed earlier? Sort of to bring it all together. If we look at the pollinators that I saw up in the canopy, the Cape Honeybee, yes, it dominates, but it's not a forest specialist species. You find it in people's gardens and in the fynbos as well. If you just want to speak strictly natural, it ventures into the fynbos a lot. Asarquina africana is a hoverfly species that you find across the continent in different biomes. Same with this hoverfly species, you find it in grassland and in fynbos. Carpenter bee across the continent, longhorn beetle, fynbos, karoo, grassland. So these insects that were quite abundant up in the canopy aren't really forest species. They're stuff from outside the forest. So it links up with what I predicted earlier, that there's spillover. But we also found some forest specialist species. So the two hoverfly species that I mentioned were described last year. The one is found in Afromontane forest from George, where I found it in canopy, up to Afromontane forest in Malawi. And there's another species that as it's larvae that live in stagnant water in old trees. So it's also sort of reliant on large old trees in the landscape. But many of these species are poorly described. We don't really have ecological information on them. That's the nature of insect science. We know very little. Something has to become a pest before there's funding for it. But what is important, I guess, as a takeaway message is that this is a two-biome interdependent landscape. It's not that we just have to conserve the forests or the fynbos. They're interacting with one another, and they're ancient in this landscape, and the ecotones, the edges, are very important. Last slide, I promise. Smaller patches, not necessarily less diverse. You have to conserve the matrix as well as the forest because of the possibility of spillover, protect the edges, and one tree species will support many insect species. Trees are like red wine. The older they get, the more insects they support. Leaf litter that they shed, the bark, the lichen, the moss on the branches, the epiphytes. An old tree in the forest is like an island on its own. You can make a career out of it. You just need to win the lotto first. Okay, thank you.
Right.